doesn't work. Okay. Yes, thank you very much, everyone, for being here. Welcome to the first Physics of Life seminar. It's a bit of an experiment. Normally, we are not in the physics department, even so it's a Physics of Life seminar series. We're normally either in life science or something else, bioengineering. But we also wanted to advertise a little bit our network. So, in case people are interested in biological physics, there's some activities and journal clubs and so on. Anyway, in case you don't know me, I'm Robert Endres, and I'm one of the directors of this network, and there are a couple other people as well. So anyway, welcome again for being here. It's my great pleasure to introduce Boris Shraiman from the KITP in Santa Barbara. He's a Susan F. Gurley Professor for Theoretical Physics and Theoretical Biology at UC Santa Barbara. And he's currently on sabbatical at the Newton Institute in Cambridge. And a little bit about his CV. So I think he got his PhD from Harvard University and then a postdoc at University of Chicago and then a bunch of other things. Bell Labs certainly, and Rutgers as well, briefly, before he came to Santa Barbara. And he won lots of prizes. He's also a member of the National Academy of Sciences in the US, so really nice stuff. And and he's really, today he's actually talking about one of his main interests, connecting mechanics of tissues with biology. That should be ideally a talk for bridging physics and biology and for being here in the physics department. So thank you for being here, and over to you.
Well, thank you so much for inviting me. And thank you to you all for coming here. And I guess I'm bringing morphogenesis to the physics department here. I suppose maybe this is something that happens to you with age, but I'm going to start in a little bit of a philosophical mode with a question of what physics contributes to biology and vice versa. Well, certainly in the last 20 years, or maybe 30 years, again, there is kind of an explosion of activity at the interface of physics and biology, with lots of physicists kind of migrating towards biology. But, biophysics has been there all along, is a very well-established field. And there is plenty of sort of biophysics in the strict sense that I will say here that contributes to biology. The contribution is really not in question. Take all of structural biology for that matter. So, of course, obvious contribution is physicists come bearing gifts, as in tools and instruments. But what I will mostly focus on is kind of a different export from physics to life sciences, which contributes now to the development of quantitative biology. And I'll be very much focused on phenomenological modeling and search for simplicity in dealing with complex biological systems. And then, of course, this whole enterprise enlarges the domain of physics altogether, introducing new models and paradigms for physicists to think about. And then, of course, there is perhaps physicists can help biologists formulate new questions that haven't been asked yet. At least to formulate questions, if not answer them. But let me start with just a little example of what you might call sensu stricto biophysics, right? The recent discovery of liquid-liquid phase separation inside the cell, underlying formation of sort of intracellular chemical factories where preferential segregation of certain biomolecules underlies function. So That's a very big recent discovery driven by physics methods and thinking, and some big prizes being awarded to the discoverers, and there will probably be more prizes still. Very active current field. But, and this is, biophysics, biophysical interactions, biophysical phenomena. But physics ideas can also be useful in different ways, and I will call it here sort of an off-label use of physics concepts. For example, if you look at the back of your hand, right, there are little hairs there, and you'll notice that they preferentially pointing in one direction. And for example, on the wing of Drosophila, okay. So is this me or is this the pointer? Did I do something? Can you see the pointer? Okay. This is me and technology. But this never fails. Okay, so there is this fly wing, and there are little hairs, and they're also all pointing in the same direction, and there's a whole story, very interesting story that can be told about how this pattern is formed. And the analogy with magnetism in physics is inescapable, right? So you might think that general interactions, general phenomena underlying the alignment of, thank you. Oh, perfect. Of these little hairs can be really informed by ideas from ferromagnetism. But I assure you, there is no magnetism involved in alignment of those little hairs. Only the ideas. It is virtually impossible for physicists, especially theoretical physicists, to give a talk about biology without talking about Darcy Thompson. And for me especially, because I just visited Dundee last Friday, and found out that that's where Darcy Thompson was. Darcy Thompson famously thought about mechanisms underlying some beautiful shapes, morphologies observed in, exhibited by found in living matter, and mechanisms that underlie it. And he also famously thought that developmental biology or morphogenesis was basically a problem in mathematics and physics. So, of course, he was thinking about morphogenesis very early on, right? This was just the dawn of genetics, and biology actually knew very little about mechanisms underlying development and morphogenesis. We've learned enormous amount since, and now, for example, with progress in developmental genetics, we know exactly what kind of a gene, if expressed in the wrong place, will convert an antenna into a leg. So literally, inappropriate expression of a single gene flips a switch on the developmental program, which turns the outgrowth here. So changes the morphology of the outgrowth from antenna into the leg, right? And we know all that. The only thing is we still have no idea of what it takes exactly to change the shape of the outgrowth from an antenna to a leg, right? But then if you think of, so the fundamental difference here is antenna or a leg is a label, right? It's a binary phenotype, either or. But when we talk about shape, this geometry, we need a quantitative description. So it's what's called the quantitative phenotype. So there is work to be done. And in fact, the kind of work that Darcy Thompson dreamt about, wouldn't it be nice to, wrong way, revisit his agenda of growth encoding form now with the knowledge of developmental genetics and cell biology. So how do genes encode geometry? That's a big question. There is some wonderful work done in UK that provides us with wonderful examples. So this group of Rika Cohen in plant biology lab has identified one particular gene misexpression of which changes the shapes of leaves. And so this gene controls cell growth and proliferation. So when this gene is non-uniformly expressed, it causes non-uniformity in growth. And then if there is more growth in the middle of the leaf, right, that causes sort of bulging out into hemisphere. And conversely, if there is more growth on the periphery, then effectively the perimeter, the margin of the leaf overgrows for the area, and then it has to form little lettuce-like crinkles. So a very direct example linking gene expression and growth and shape. And these ideas have been taken back to a lab where it is possible to sort of encode basically polymer elastomers in such a way that upon curing, these elastomers expand differentially, and then one can sort of make hemispheres, or, well, anyway, one can produce sort of non-trivial shapes by programming an expansion within the synthetic material. So bio-inspired shapes. Well, we can start thinking more about the relation of growth and form, even in the sort of simplest context of sheets. And so forming three-dimensional shapes, right? We know from Gauss that any three-dimensional surface can be mapped back into a flat disk, not any, so topological disk, can be mapped into a flat disk by conformal transformation. So conversely, that means that we can take this flat disk and grow it non-uniformly and generate this arbitrary 3D shape. So the only thing is that in biology, we often observe not just isotropic growth, but anisotropic growth, right? So instead of diagonal metric, you have sort of full symmetric matrix. So instead of encoding locally one number, you have three numbers to play with, right? So you can certainly grow anything, and in infinitely many ways. And now you can ask, well, now that there are many ways of generating the same shape by growth, could it be that some ways are better than other ways? And, well, you can start thinking about it, and you realize that there's a trade-off between uniformity of growth and anisotropy of growth. And, for example, by allowing anisotropic growth, you can achieve the same shape in a more uniform way, right? So if you think that specifying spatial pattern of growth involves so more information, then growing with sort of more uniform pattern might be in some sense easier, right? And as a physicist, you can have fun formulating so an action-like principle for growth and deciding that some patterns might be in some sense better than others. So the only problem with that is optimization here is not unconstrained. And it's constrained by, if you like, biological hardware or wetware. So we can't just engage in this theorizing without understanding more about the underlying mechanisms. And at this point, I have to switch from my philosophical introduction to actually saying something hopefully of substance. So I'm going to continue to talk about form and its relation to mechanics. But I'm also going to start by pointing out that differential growth, with due respect for Darcy Thompson, is not the only thing that biology has on hand in generating patterns. And I'll give you another example here. And this time we're going to begin with actual data. So right now you're looking at a snapshot of taken through a light sheet microscope, and you're looking at Drosophila embryo, which has the overall shape of an ellipsoid, and these dots here are nuclei. So there are lots of cells lining the surface of this ellipsoid. We'll stick to these two-dimensional sheets for now. And the color here actually corresponds to, so yellow here is an expression of some particular gene. It's not very important for us which one. I'm just illustrating that all the nuclei, of course, are genetically identical, but some are different from others by virtue of expressing different subset of genes. One of them is visualized here. And now I'm going to hit return and turn the snapshot into a movie. So at first you see the emergence of these stripes. Stop. So the pattern of gene expression emerged. And the next thing you saw was a rapid rearrangement of cells on the surface. And I'm going to try to play this again. and maybe even stop in motion. So now up. The first thing that happens is cells on so the belly of the embryo dive in to make an inner tube. That's the first step of gastrulation. And the next thing that is going to happen is cells from here will so start moving from the ventral side onto the dorsal side. And that's what you're going to see next. If I can make this work, and I can't. I'm very sorry about this. All right. Okay, never mind. So this imaging was done by Sebastian Streikan, who is now junior faculty at UCSB. He did it when he was a postdoc. And to better see what's happening with the motion of cells on the surface of the embryo, he likes unrolling the embryo computationally onto a square, kind of Mercator projector of the embryo, so that anterior is here, posterior is there. This is the this is the belly, ventral furrow is forming, and the cells from here, right? There's this flow of cells onto the dorsal side that you see over here, and please play. Okay. So this process is known by the name of germ band extension or convergent extension. And the reason it's called convergent extension is because it's basically hyperbolic kind of flow. So here Sebastian so carried out PIV and basically mapped out the velocity field. And you see that what's happening is there is a kind of hyperbolic convergence and divergence point at kind of dorsal midpoint. And then there is another hyperbolic fixed point which got cut into two in this picture, corresponding to ventral midpoint, and the whole thing, of course, is happening on the ellipsoid topology of a sphere. You have two hyperbolic fixed points. You also need to have four elliptic fixed points, and they're all there. And we have a nice flow, except it's a flow of cells. And we would like to do sort of hydrodynamics of tissues, but the question is, how do we describe the flow of tissue? What's the correct description? And the first question is the tissue fluid or solid? People are arguing over it. And obviously, this flow of cells, flow within the tissue, is goes beyond textbook continuum mechanics, because it may look like a flow, but it is driven by internal activity. There are myosin motors, for example, in this tissue that are driving the flow from driving cell rearrangements from inside. And in addition, there is also internal control, because cellular processes are regulated. In particular, they're regulated by differential expression of genes. And at the very beginning, I showed you patterns of gene expression. I only showed you one gene, one pattern, but there are more genes and more patterns. So how do we begin to think about it? So let's dive in. So we actually have these very beautiful imaging of epithelial tissues, right? So if instead of looking in nuclei, you look at cell interfaces, you find that you observe this sort of very nice geometric tiling of the surface. And you can zoom in and see it even better. And then the question is, if we could actually zoom in and see inside, what will we see? Well, what's mechanically most relevant inside is actomyosin cortex, which is formed on the inner side of cell interfaces and consists of actin and myosin. So actin, basically structural polymer, and myosins are little motors that can walk along here and cause the bundle to contract. So this is inside the cell, but these cortical bundles are welded together by adhesion molecules, which traverse the membrane, form bridges like that, and essentially form sort of a tissue-wide mechanical network. So, I just introduced three important molecular players, but of course, in reality, there are dozens, if not hundreds more. And the question is, when we dive into this molecular description of cellular processes, where do we stop? And so we can go deeper, but our approach is going to be actually look for a simpler description, right? So rather than add more relevant players, we're going to try to sort of sublimate the essence of mechanical activity here. And that brings us to a type of vertex models where we parameterize the geometry of cells just by specifying vertices in a polygonal tiling. And then we're going to assume that mechanically relevant activity is associated, can be parameterized by tension in these interfaces and pressures, except we're going to imagine that these tensions and pressures can be independently controlled. So we're going to describe elasticity here in a kind of minimal way, which allows us maximal flexibility. So rather than pretending to know exactly what's generating the tension interfaces, we're just going to say that, okay, what's important are sort of the changes in sort of geometric changes, right? Extension of edges. And there is a conjugate variable associated with length, that's the tension, conjugate variable associated with area, that's the pressure. And we then imagine that these tensions are generated by, in some controlled way, by actomyosin cortex. But what our elastic energy here describes is sort of fast dynamics, rapid relaxation to mechanical equilibrium, right? So minimizing this energy means that tensions, and sorry, I'm going to simplify things a little bit by saying that let's forget about possible variation of pressure between cells and just say that then negligible, and pressure is there just to prevent the global collapse of this tensionized network into a little lump, right? And in that approximation, mechanical stability means that tensions, so the sum of tensions at each vertex has to be 0. So just force balance. Well, so amusingly, even this sort of simplest bit of physics is constraining and informative. And I'm going to convince you that this is so. So for each vertex, we have this force balance. And when you have 3 vectors adding to a 0, you can turn them around and make a triangle. And right, because each edge contributes to 2 vertices, it contributes to 2 triangles. Edges are shared by triangles. So these triangles basically have to tile the plane. So we have a tension net. We have a triangulation of a dual space, tension space. And if you stare at this for a couple of hours, you'll realize that the fact that triangles tile the plane implies a geometric constraint on its angles. And in a very simple form, you define some pairs of angles, ratio of sines, product around the plaquette. This has to be equal to 1. It just follows from the law of the sines in 5 minutes. But amusingly, this follows immediately from the assumption of force balance, right? And we can start testing it. So, because remember, you can get a hold of these beautiful images of cells interfaces. You can go and estimate all the angles, and you can do it in different tissues. And you can start testing whether this assumption underlying, or rather, relation that follows the assumption of mechanical equilibrium is actually correct. And what you find is that, of course, I said this product is equal to 1. We took a logarithm, so now it should be 0. Nothing is exact. There is some distribution. But what you can test is whether there is a tendency for this log chi to be 0, which is statistically significant in some suitably scrambled null ensemble. And you find that in some tissues, there is a strong tendency, and in some other tissues, there isn't a statistical significant no, this is not statistically significant. And that basically tells you that it is probably, so this is, one particular larval tissue. It's okay to push forward building a model based on force balance in this tissue, but not in that one. So there is a non-trivial statement, and you got it from very few assumptions. But we're not done yet, because I just showed you. Yeah, please.
It's not a good approximation.
Yeah.
Correlate to the flows. Let's say in your case, what you showed me is these flows are extremely slow and sort of fast equilibrium. And with the wing disk, can you see some noticeable difference in terms of the dynamics? Is it much more dynamic?
Well, actually, what's happening is it's cell morphology. So that wing disk is what's called pseudostratified epithelium, where nuclei are packed into sort of multiple layers, and there's actually a lot of distortion going on. But we'll come back to the question of dynamics in a moment. But I'm actually doing very badly on time. Okay, so for a given tiling of cells, I argued we can construct a triangulation. Now, going back, you discover that if you have a triangulation, there are many possible cell tilings which correspond to it. And that's basically because you can show that there are deformations which preserve all the angles of the triangulation. So preserve angles, so we call them isogonal, so there are these isogonal soft modes. And lo and behold, this is another prediction that you expect to find this particular mode of cell deformation, which tends to preserve local angles. And then we can go back to experimental data and look in particular at the time just preceding this first step of gastrulation, when cells are moving, so converging onto ventral midline, and one observes a lot of sort of strong local deformation of cells. And if you analyze that, you find that 80% of the variance in this deformation is associated, can be accounted for by this isogonal deformation. Okay, so hopefully I convinced you that 2D geometry of cells is constrained by mechanical equilibrium, but one can push this further and say, well, if mechanical equilibrium is so constraining, maybe we can use observed geometry to actually infer what tensions there are inside cells, and that's very useful because they're very hard to measure directly. And lo and behold, you can convince yourself that you have enough observables to infer, for example, edge tensions. Well, fine. So there is an algorithm. You can run it on your favorite image of epithelial tissue, and it spits out some predictions of color-coded here of tension. When so applied to one particular image, which you can segment. So how do we test this, right? It's very difficult to measure tensions directly. But in this image that comes from Tamali Kui's lab, they actually imaged myosin, right? That molecular motor that's generating tension in the first place. It's in green. And it's not completely ridiculous to think that tension in this filament is actually proportional to myosin that we see here. And then we can take our inference and correlate with local intensity of myosin and see how much correlation there is. And again, this is low accuracy kind of analysis, but there's a lot of data. So you can look at kind of statistical distribution. So here is a cumulative distribution function of the correlation coefficient. And the median correlation that you find in the data is 0.4, right? And if you scramble the angles before you analyze them, you'll find 0. So this is sort of hugely statistically significant, but now the correlation coefficient is 0.4. So the question is that good or is that bad? There's a correlation, but there's not a lot of it. Now, I'm going to argue that actually it's really surprising that there is that much correlation. Because I'm showing you one snapshot here. But in reality, this is a movie. And it is actually pretty amazing that there is a proximate force balance in these individual snapshots. So we are dealing somehow with this very peculiar material, this active material in our tissue, which can both be in a proximate force balance and also flow, rearrange itself. So let's think a little more about modeling now, going from statics to dynamics. And I said that in the beginning that we really want to do away with constitutive relations because cytoskeleton is less a Hookean spring and more a muscle. And my muscle can generate different tensions quite independently of extension, right? There's no constitutive relation. But nevertheless, it's useful to think of a Hookean spring here, but a spring with internal length that is itself a dynamical variable. And what's happening is that depending on how much myosin there is in this filament and how much tension is applied to the filament, it can either contract if there is more myosin than external tension, or it will start slipping and extending if there's too much tension. And if there is just the right amount of myosin for a given external tension, it will remain at stall. So this is how we can begin thinking about dynamics. And this sort of model, although now I'm only explaining it on the level of a single little filament, if you extend it to a description of the tissue, it will not surprise you. It effectively corresponds to viscoelasticity, right? So elastic response at short times and sort of fluid-like behavior at long times. Well, and that is a little surprising because, fluid behavior at long times, I'm standing here, and there's epithelium on my arm, and it's really not dripping away, right? So it isn't really fluid. It is somehow solid. It has to be able to find mechanical equilibrium back again. And let's think about this for a moment. So to balance its internal forces, tensions have to make a triangulation. And I just argued that in equilibrium, at stall, local tension is controlled by local level of myosin. So that basically means that myosins on edges have to effectively triangulate space, right? So they have to be correlated on different edges. So the question is, how do you coordinate myosin, which is controlled locally, so that you achieve global force balance? Well, think about this for a moment, and you realize, okay, the simplest mechanism is a kind of mechanical feedback where myosin recruitment rate is controlled by a rate of strain. It's a very simple idea, just saying that if on a given interface you don't have enough myosin, it's going to extend, right? And this positive rate of strain then is a signal, please send more myosin. Conversely, if there's too much myosin, it's contracting, right? Negative rate of strain is a signal, please remove some of the myosin. So that's, you know, as a physicist or a mathematician, that's sort of the simplest way you would solve the problem. But it's just a hypothesis. So Sebastian, or rather I should say it was just a hypothesis, but Sebastian Shraiman and his students did experiments, again working in fly embryo, so specifically looking for the effect of strain, or rather rate of strain, on myosin recruitment, and they basically discovered that there actually is a pretty clear dependence of this sort, and in fact nailed the prefactor here. I mean, obviously it's not it's not just a constant, but you know, alpha here is of order 2. So the good thing about simple ideas is that they're testable and can sometimes be right. So let's put things together a little bit. So we have a description of this epithelial cell mechanics on 3 different timescales. On the fastest timescale, the response is elastic. On the intermediate timescale, right, myosin starts walking and slipping. That's sort of active viscoelasticity. And then on the slowest timescale, we have a recruitment of myosin with dynamic feedback, but also sort of biological control of myosin activity. But in particular, we have this mechanism of always restoring rigidity in the tissue at long time scale, right? So from the point of view of textbook continuum mechanics, it's a somewhat unusual situation where on shorter time scale, you can have fluidity, but on longest time scale, you recover rigidity. So, you know, we call it sort of an active solid behavior. And that gets us into thinking about how, so back to morphogenesis, how biology can then control mechanical activity in order to move cells about, change the shape of tissue. And the idea is that right, morphogenesis is happening on long time scale. It's fast in flies, but it's still relatively slow. And we propose that the way of thinking about it is not really a flow in response to forces as in hydrodynamics. Yeah, I have some scientific background in fluid mechanics, so it's not like that. Rather, it is, we think, a controlled rearrangement of the force balance. And I'm going to give you a hand-waving illustration of this using my hand. When I move my arm, acceleration is irrelevant. It is basically in mechanical equilibrium at any instant of time. Right? I am just reconfiguring the force balance of these redundant muscles that are controlling my arm in such a way that I can wave my hand at will here. And that's the way we'd like you to think about flow of tissue. Okay, again, so this is just a theory, but we have data, and we can go to data and see how far we can get with this theory in understanding the data. So back to that convergent extension flow on the surface of the fly embryo that Sebastian Shraiman is quantified. I showed you these pictures, but Sebastian also measured more. He also can visualize myosin distribution, and so you see these myosin filaments that are quite anisotropic, and so because they're anisotropic, you really want to describe them by so magnitude, but also direction. So you define not a scalar quantity, but a tensor quantity. And it's defined on the whole embryo. So we have this measured, wrong. We have this measured myosin tensor, and I argued before that myosin is generating the stress. So let's just say that stress tensor is proportional to myosin tensor, and the divergence of stress is a local force. And anyway, so you and expect that tissue flow velocity effectively, I said viscoelasticity, right? So effectively satisfies Stokes' equation with these two effective viscosities as the parameters, and this is measured. It's just an equation that you can solve on the surface of the embryo, compute this velocity from this equation, and compare with experimental observations. This is what Sebastian Shraiman and Nick Knoll, my student, did. And I guess one of these is measured, and the other one is predicted, or vice versa. And this is a difference. And quite remarkably, with just 3 global parameters, global meaning uniform on the surface of the embryo, you can achieve what I'm going to claim a rather ridiculous accuracy in description. To the extent that you then start looking at regions where you have a disagreement, and you can start asking, why is this happening here now? And this turns out to be useful. So,
so 3 parameters will fit to get this?
Yeah, global parameters. So, so it's kind of a curious situation, because this is I think, much better than anyone had any right to expect. And on the other hand, it's nothing at all, because so you have this description, but no ability to predict, right? Because all we can do is relate one snapshot of myosin to a velocity field at the same time. But we'd like, we're in the physics department. We'd like to have an equation of motion. We'd like to predict what's happening in the next moment of time. And how do we do that, right? We'd really like to be able to predict dynamics of myosin. So there is more work that has to be done. And now I'll try to tell you a little bit about that, even though I think I only have 5 minutes. Clearly, in order to be able to predict what's happening, we need to understand something about control of myosin, so biological control of myosin. This is happening on the scale of cells. So I just paraded this continuum model for you. Now we have to take it back and talk about cells again. Luckily, in the meantime, Sebastian has produced even better imaging, and his collaborators at Princeton, Thomas Stern in particular, did a rather amazing job at machine learning assisted segmentation, and so all the cells here are tracked and color-coded so that you can see where they're going later, and you can look at cell shapes and what they do as a function of time, and you can decompose this flow into and so the associated strains and rate of strains. You can decompose them into two processes, so deformation of cell shapes and cellular rearrangement, which has a form of this neighbor exchange known as a T1 process, right? Where these two cells that weren't neighbors before come together and become neighbors. And in the process, it's sort of mesoscale convergent extension. Two cells come together and the other two cells go apart. Fritjof Brown's postdoc at KITP and Nicholas Clausen, a graduate student that Sebastian and I share, took over and basically took advantage of having this sort of beautifully characterized cell geometry over time, and actually ran this tension inference analysis on all these tilings, and so produced the following picture of the process. So initially you have this cell array, which is reasonably sort of well-ordered, not too far from hexagonal, but not exhibiting any particularly clear pattern of tension. And the next thing that happens is a tension pattern emerges with sort of alternating, approximately alternating high and low tension edges. And the next thing that happens is these junctions under high tensions contract. and undergo these neighbor exchange T1 transitions. And at the end of the day, what they observed was this patch of tissue actually underwent convergent extension, and due to coordinated local neighbor exchanges. And curiously, while this was, so as a result of this process, cell array got very disordered. So initially, it was nearly, so not too far from hexagonal, but now towards the end, cell shapes are so statistically indistinguishable from what you would get in sort of random tessellation, in sort of Poisson lattice. So, so based on this, Fritjof and Nicholas put together a model which now is actually predictive of the dynamics. And you can understand what's happening there simply by saying that in addition to that negative feedback on myosin that was driving towards mechanical equilibrium, now there is also positive feedback which is driving it away from equilibrium. And then all you need is sort of modulation in this feedback so that it doesn't kick in uniformly on the whole embryo, but sort of more on the lateral sides and less on the dorsal side, right? Consistent with genetic patterns or patterns of gene expression that we know. So, and another curious conclusion was that there seems to be information in cellular order. So anisotropy and sort of proximity to an ordered lattice appears to encode information. So that, at least in simulations, the overall extent of convergent extension correlates with the amount of order, or if you like, disorder in the initial condition. So these are, again, falsifiable predictions. And we haven't tested them yet. But my overall message is I hope I sort of gave you a taste of sort of a thought process where you build phenomenological, so simple phenomenological models in such a way that they are testable along the way. And then you sort of add little bricks as you go along, but in such a way that you can tether this theoretical edifice at many points to actual data. So, simple models can go a long way, and it actually is possible to make predictions in biology. So what happened to all the complexity? And this is something that really keeps me awake many nights. So remember how I bragged about that simple model with 3 parameters that explained data? So it's really a very strange thing because so Sebastian measured myosin, right? And myosin, of course, is very important. It's the motor. but also remember, there's actin and cadherin and 300 other important proteins, and many of these genes, if you mess up with them, it's going to break everything. So what happened to them? Why is it that knowing in some quantitative detail just one observable, tensor, why is it enough? Well, I don't really have an answer. But one thing that comes to mind is that in this complexity, there's structure. That there is some sort of self-organization going on, which then correlates how much myosin and how much cadherin and how much of everything else you get, so that if you know just one, in a way you already know where on some manifold of, right? So there's some constraint in this horribly high-dimensional space where the cell, the tissue, has to live. And knowing just not too many things is enough to tell you where the cell is, right? That there is some sort of self-organizing principle hiding there and defining the structure. And discovering that principle is I think, worth spending time on. Most important slide. I think I already introduced most of these names. So Fritjof is a postdoc at KITP. He and Nicholas worked on this sort of last story, so cellular description of convergent extension. Nick Normadov Mani and it's Hemskirk, so previous generation of students, postdocs, and collaborators at KTP that we worked on this formulation of the active tension network model. And I mentioned Sebastian Strachan a lot. He is the source of all our data and a lot of our analysis. And we've also enjoyed and benefited from tremendously from a collaboration with Eric Wishhouse at Princeton and his student, Matt Lefebvre. Thank you very much for listening.
For this lovely talk, nice mixture of philosophy and quantitative biology.
I went a little long in philosophy.
Any questions? It's quite late, but there's no one afterwards here in this room, so we have a bit of time. I guess,
Yeah, please.
So again, back to this kind of data I could explain with 3 parameters. So you measured myosin, and what was being explained was the
Flow field.
flow field of the cell shapes and cell movement.
Well, not cell shapes in a particular case. It's already coarse grained, so it's just the flow field. Yeah, just PIV data. Then of course what I didn't show you is so that's one particular point in the movie. This is done as for each time slice. Sometimes the agreement is not as good as this, and for example, when there is this ventral furrow formation, this description loses accuracy because we're not allowing cells to go inside, right? So then you say, okay, well, we can introduce so an absorbing boundary condition and introduce more parameters, and then of course you recover the accuracy at the expense of more parameters. So in that way, it's not surprising. But kind of deviations from the model become sort of useful indicators. There was another question somewhere. Yeah.
Follow-up question. So you said the accuracy wasn't based on the color coding. Not very good at the posterior.
Yeah.
The two ends basically.
Yeah. So in that particular case.
Imaging problems.
Yeah, so that particular thing is mostly imaging. Yeah, so other right. So remember there are these elliptic points, so the kind of vortices going. It is very easy to make an error in locating the vertex. So that's sort of another area where you get high errors. Yes, please.
My question is more about the philosophical point of view. If it's not related to this specific case, I mean, when you assume there is a kind of, let's say, reducible space about a phenomenon, it means that your mechanistic description is so redundant that you can easily map it back to some lower dimensional space by less amounts of. So, isn't it mean if we assume we are looking for this reduction manifold in any biological model, we are assuming our model are so much, I don't know, is it right to use redundant and bad that we are looking for? Because in your case, it means that number of degree of freedom is too much, so you can just reduce it to some smaller space.
Well, it's not, in this particular case, it's not a matter of the model being redundant. The model is already low dimensional. With due respect, what I'm trying to say is it's not redundancy. It is self-organization of an actual physical, if you like, or biological process, right? So, and let me give a trivial example. Talking about mechanical equilibrium here, right? And so we can talk about manifold of mechanical equilibrium states. Very simple geometric picture. Any triangulation is mechanical equilibrium. There are lots of them, right? But not every configuration of cells can be in equilibrium, right? There are geometric constraints, right? So actual space is larger than the manifold of force-balanced states, right? But I'm just giving sort of a mathematical and geometric example so that it's easier for us to think about it. And then from there is this sort of giant leap into unknown, where we start talking about how perhaps there is a feedback that is organizing not just myosin, right? I gave you sort of a very specific prescription that has been confirmed experimentally of how to determine local level of myosin so as to establish force balance. But perhaps there is a similar thing that controls not just myosin, but actin and everybody else, right? So then we have, and again, mathematically, we can sit down and propose how it could be implemented, right? It's a sort of mathematically viable scenario which will define a manifold of cell states where everything is going to be correlated by virtue of this dynamics. But we have to test it. We have to find the right description.
So in the
beginning also you showed, these images, these movies of the embryo, and it was still a syncytium, right, of nuclea, I think you mentioned, right? And later,
Yeah, so
this is transition, I mean, obviously it needs, you talk about the junctions.
Yeah.
Rearrangement. So.
Yeah. So, yeah, well, very good. I was certainly trying to go fast, even though I was fumbling with the movie. Initially, and this is unique to a fruit fly that's in a big hurry to develop, is just a syncytium, and there are no actual cells, even though what's happening is cortical cytoskeleton gets built even before the cells are actually fully closed. But sometime so just before this ventral furrow formation, the cells actually fully cellularize. I mean, fully form. And from that point on, there is the proper epithelium. So we actually see an amusing signature that corresponds to, roughly coincides with cellularization. So remember how I had these two viscosities, and one was shear and another one was effectively bulk? And so early on, we basically see that these two viscosities are somewhat comparable, and later on, one of them, right, associated with compressibility, becomes very high. So effectively, tissue becomes incompressible, and one can fantasize that having closed cells has something to do with it, but it's a fantasy because the water can still be exchanged.
Yeah, thanks for the talk. I'm learning about feedback from myazine. What's the time scale of that feedback?
Minutes?
Do you know how it's implemented?
Oh, I'd love to know that. It's non-trivial, right? Because it has to respond to not to strain, but to rate of strain. So it's a little delicate.
One more question. So you talked about the contraction of the cortex, effectively the myosin in the cortex to lead to these rearrangements. You showed the flows and so on. But of course, then you have on top of that these bigger flows. You said, you can't describe the gastrulation event, the inward folding and so on.
Yeah.
So there's of course other stuff happening, and I was wondering,
absolutely.
Controlling that again. I mean, how?
So Eric Wischhaus and his students have described that in students and postdocs have described it in great detail early on. It's actually driven by a different pool of myosin. So medial myosin on the basically apical surface of the cell. And it is causing this local contraction. And one can basically have mutants where it does not happen altogether. And then, interestingly, this germ band extension, the convergent extension flow, will still proceed in those mutants, but will be quantitatively different. And that's of great interest to us because there are some fairly subtle changes in the situations that they do not produce kind of a quantitative binary phenotype, right? Because there's still extension, but they produce a quantitative phenotype. They're rather slight, well, not even so slight, but reproducible differences. And it's those kinds of mutants that are a real gold mine for us now, because we're trying to be more quantitative about describing it.
Yes, and thank you again for your fantastic talk.